Okay, so today we're going to talk about amino acids, and we're going to talk about the different things to do with amino acids. So this is suitable for high school students in IB, IGCSE, and just high school biology in general. So starting off, what are amino acids? So amino acids are the monomers of proteins. So monomers means the building blocks of proteins. So amino acids are what proteins are basically made of. Okay, so amino acids differ in their variable side chain. So as you can see here in the structure of an amino acid, the basic structure of an amino acid, there's this R group over here. So this R group is what differs in all of the different amino acids, and the R group is what gives it its specific biological properties. So let's look at the basic structure of amino acids. So we have an amine group, which is this NH2. Then we have this carboxyl group, which is C double bonded to oxygen and then bonded to an OH. So COOH is how you can remember it. All right, so this is the basic structure of amino acids. And like we said, they all differ in their R group, which is the side chain. Okay, so when we look at the types of amino acids, we basically have three types of amino acids. So first of all, we have essential amino acids. They're essential, meaning that we need them in our diet because they are not produced by the body. So because we don't produce them, we need to get them from the food we eat. That's why they're called essential. Next type is non-essential. So this is basically the complete opposite. These amino acids, we produce them naturally, so we therefore don't need them in our diet. All right, then the third type of amino acid we have is conditional amino acids. So basically, conditional amino acids are produced by the body, okay, but they're usually produced in lower amounts. Then during certain times, for example, pregnancy, we need more of these amino acids, okay? So they become essential, basically. All right, so to recap, conditional amino acids, they're produced by the body but in low amounts, okay? Then during certain times, for example, pregnancy, we need more of them, so they become essential. So that means that we need to take them in our diet. Yeah? Now, when we have a shortage of amino acids, we get less production of certain proteins. All right, because we know that amino acids are the building blocks of proteins, right? So when we have shortages in certain amino acids, we get shortages in certain proteins, okay? And when we have that, we get protein deficiency. So that's why when you have certain diets, people who follow certain diets, they may have some sort of protein deficiencies because they're not consuming certain essential amino acids. Okay, now let's look at how these monomers of amino acids are actually combined together. So they are combined together in a reaction called condensation. So condensation means loss of water. All right. So over here we have two amino acids that have been drawn. All right. You can see here NCC, NCC. Now what happens is the oxygen of the carboxyl group and one of the hydrogens of the amine group combine. So we have two hydrogens and one oxygen, right? So that gives us water. That's why condensation is loss of water. So when they combine, okay, there we get a peptide bond. Okay, so let's look at what exactly is happening here. You can see here we have the carbon over here and this carbon over here, right? So this hydrogen is lost, so that's why there's no hydrogen over here. And then when you look at this amine group, this hydrogen is also lost, so you can see there's no hydrogen up there, right? So this carbon is combining with this nitrogen, and in the process, water is being released. So here we have our peptide bond plus water. Okay. Now, if we want to do the opposite way, if we want to convert the like dipeptide back into the amino acids, we're gonna add water, okay, and we're gonna get a hydrolysis reaction. Okay, so condensation is loss of water, hydrolysis is additional water. Okay, so now long chains of amino acids are called polypeptides. Poly meaning many. Okay, so like I said, they get broken down via hydrolysis. Now, like I had said before, the variable side group, this R group over here, it is what gives the amino acid its specific properties, all right? Like its specific chemical properties. So, for example, non-polar or polar, charged, etc. So when the polypeptide group is folding, it's going to fold according to these distinct chemical properties of the side chain. Okay? And as a result, we can get a lot of various types of proteins. So here are the proteins and their functions. So we have proteins that have a structural function like collagen, a transport function like protein channels, proteins in the immune system like antibodies, hormones like insulin, sensation like rhodopsin, movement, actin and myosin proteins, enzymes like amylase. Alright, now lastly, let's talk about denaturation. So denaturation is what occurs when there is a structural change in a protein, okay, structural change, and that causes the protein to lose its biological activities, because its biological activities is dependent on how the protein has folded. So when we change this structure of the protein, then we lose that biological activity. So denaturation of proteins is caused by temperature and pH. So high levels of temperature, high temperatures, basically break hydrogen bonds. Okay, and temperature, the optimum temperature of proteins varies. The optimum temperature of protein varies, but usually like the body proteins is going to be like around 37 degrees Celsius. That's like normal body temperature, right? So that's why when like you have a fever where your temperature becomes like really high, one of the dangers is it can cause denaturation of proteins. Okay, now the next thing is pH. Okay, so pH alters the charge, which then alters solubility and the overall shape of the protein. So that brings us to this concept of Zwitterions. Okay, so a Zwitterion is basically when you have this neutral molecule that contains both positively and negatively charged regions. Okay, so here is a Zwitterion. The amine group here is protein is positively charged. You know how it was NH2 and now it's NH3 plus. And this carboxyl group is COO minus. So that's negative, okay? Then we have here when both groups are protonated, okay, with the hydrogen, then both groups deprotonated, lack of the hydrogens, okay? You can see here, this hydrogen is being added in, here the hydrogen is being removed, okay? Just the main concept that you need to keep in mind is that a Zwitterion is a neutral molecule that has both positively and negatively charged groups. And you see here it's neutral because this positive charge is balancing out this negative charge, which makes it neutral. Right? So when we alter the pH of the protein, that affects basically these charged regions, which then affects the solubility and the overall shape. It affects solubility because like for example, you say polar molecules dissolve in polar molecules and non-polar dissolve in non-polar, sorry, not molecules, polar groups dissolve in polar solvents, and then non-polar groups dissolve in non-polar solvents. So that's kind of like how it ties in. Then here, different enzymes have different optimum pHs, just like how different enzymes have different optimum temperatures. So for example, like the stomach proteins will function at a low pH, yeah, like an acidic pH, because the stomach is acidic. Yeah, so that's basically some of the key concepts that you need to keep in mind about amino acids. And now let's do a little bit of practice. Okay, so here I'm using TutorLink's quiz generator. I put the subject as biology and the topic is amino acids. So which of the following groups are always present in every amino acid? So we know that's going to be the amine group, the carboxyl group, the hydrogen atom, and the variable R group, right? State the name of the covalent bond formed between two amino acids during protein synthesis. So we said that's the peptide bond. Outline the general structure of an amino acid, identifying its 4 key components. So for this sort of question, it would be better to draw the structure, but we know that it's going to be the amino group, the NH2. Then we're going to have the hydrogen atom and the R group, and then the carboxyl group, right? Which chemical reaction is responsible for linking individual amino acids to form a polypeptide chain? So we know that's a condensation reaction. Then distinguish between the amino group and the carboxyl group found in an amino acid. So this question, you just need to describe the structures. Amino group having 2 hydrogens and the carboxyl group having a double bond to oxygen and then an OH group. Explain how a dipeptide is formed from 2 amino acids, detailing the type of chemical reaction and the molecule that is produced as a byproduct. So in this case, you would just describe basically this condensation reaction, where the hydrogen of the amine group combines with the oxygen of the carboxyl group to form water. You just have to draw the structure and explain that. What is the primary role of the R group in determining the specific characteristics of an amino acid? Like I said, it will just define the unique chemical properties and the identity of the amino acid. State the most significant primary role of amino acids in a biological context. So we know that amino acids are the building blocks of proteins. Yeah, basically. Then describe the process of hydrolysis. That would basically just be saying that the addition of water, okay, you can imagine here in our dipeptide, if we add water, which is H2O, right, we know that this OH will come and bind here and this hydrogen will come and bind here, right? So that would cause the molecule to hydrolyze. Then when a polypeptide undergoes hydrolysis, which type of covalent bond is specifically cleaved? So we know that's our peptide bond. Okay, so let's just look at the results. I didn't put the answers there, but let's see, this is correct. Here, peptide bond, like we said. Here, the general structure, the central carbon atom bonded to the amino group, the carboxyl group, the hydrogen group, and the R group. This one we did. Here, yeah, like we said, the amino group has 2 nitrogens and the carboxyl group has 2 oxygens. Double bonded to oxygen and an OH. But you see, this question is 2 marks. Okay, this working is just to help you understand better. Over here, the same reaction we described, the condensation reaction. Here we said they're the building blocks of proteins, right? This is the process of hydrolysis. And yeah, so that's it for the amino acids. If you want to practice more using the quiz generator, you can come here to learntutorlink.com/quizlab, okay? and you can save your quizzes as well so that you can review them later. Okay, and if you're having any problems with biology or any other high school curriculum subject, feel free to come over here to learntutorling.com and find a tutor that suits your needs. Or otherwise, if you do not want to work with a tutor, you can always come here and use the quiz lab generator. Thank you.